Okay, I think we can get started. Maybe people will be joining as we start, but I'd like to welcome you to the Lithium webinar series. This particular series is going to focus on sample preparation for the light sheet microscope. And as you know, we are lucky to be equipped with the light sheet microscope, and we're trying to encourage people to come use the microscope. And so this set of webinars will help in sort of orienting us as to how to prepare our samples and what kinds of applications there are to this type of microscopy. So today I'd like to welcome Dr. Ulrich Kubitscheck from the University of Bonn, along with his PhD student, Dr. Juan Eduardo Rodriguez. So we'll hear from both of them about light sheet microscopy. And we're very excited to see the applications that they use. So just for a little background, Dr. Kubitscheck is a physicist who got his PhD degree from the University of Bremen and his postdoctoral training at the Weizmann Institute of Science in Israel, and then at the University of Munster in Germany. He was appointed at the Institute of Physics and Biophysics, and in a few years became a full professor there in the biophysical chemistry at the University of Bonn, from where he has made significant research progress in dopaminergic system development, visualizing mobility state transitions and RNA trajectories by sensitive single molecule feedback tracking, and nuclear export of single native mRNA molecules observed by light sheet fluorescence microscopy, just to name a few. Dr. Kubitscheck has been developing and building applications in light sheet systems, and actually he built the light sheet system that's located at the University of Chile. Dr. Juan Eduardo Rodriguez received his bachelor's degree in engineering sciences from the University of Concepcion in Chile. From there, he also received his professional degree in electronic civil engineering and his master's degree in engineering, graduating both with maximum distinction. He has gained professional experience working on several labs, such as the Lab of Scientific Image Analysis at the University of Chile, at Columbia University in the USA, and at the University of Waterloo in Canada, for which he received a scholarship from the Emerging Leaders in the Americas program. So among other awards, he also has a scholarship from CONICET and the German Academic Exchange Service to work on his PhD. And since 2017, he's been in Dr. Kubitscheck's lab, and I think he's about to receive his PhD degree. And so he's been focused on light sheet microscopy, utilizing clarity and expansion techniques. So we're really lucky to have these experts joining us today, but also we have some future plans with them. At the end of the year, or maybe January, we plan to hold a workshop that's going to be a week-long workshop where you can come work with them and try out these techniques. So please stay posted for those announcements. And also in preparation for that, you can come to our workshops that we're planning. So June 1, there will be another seminar where we'll start to work with clarity or prepare to work with clarity, and you can sign up for a hands-on session. So please come get started. So, okay, with that introduction, I'll hand it over to Drs. Kubitscheck and Rodriguez to present their seminar called The Transparent Brain, Light Sheet Microscopy of Expanded Neuronal Tissue and Brain Organoids.
Yeah, hello, everybody. Welcome to our presentation here. So in Bonn, we have currently 4 p.m. and a serious thunderstorm is coming up. So I hope our voices will be understandable throughout the talks. And if there is any interruption, please let us know. Okay, so let's start immediately. First, Alenka, thank you very much for the kind invitation and the kind introduction. And I hope that what we will tell you will be interesting to you. So we will present light sheet microscopy as a technique to study brain tissue in two versions, and first in the version of brain organoids, and then in the form of mouse brain sections. And I will start out giving an introduction. Why light sheet microscopy? Why should we use light sheet microscopy? What is the special features, and what can be done in order to even further improve the technique and especially increase the sample treatment and improve the sample treatment and increase the resolution. And in the second part, Juan will talk about applications, the brain organoids and mouse brain sections. Okay, so here, let me, whoops. I guess you can see my cursor, yeah? Okay, so in normal fluorescence microscopy, we illuminate an extended sample with epi-illumination, and we illuminate it usually from the top or from the bottom, but we illuminate the complete sample. Although actually, usually, we only are interested in the fluorescence that is occurring in the focal plane. But the fact that the illumination passes throughout the sample seriously deteriorates the contrast and the resolution. I will demonstrate this at this very trivial sample here, which just comprises fluorescent molecule in a solution. And when we image that with normal fluorescence microscopy, we see something like this, namely more or less nothing. We see a little bit dirt in intermediate planes, and we see, in this case, some particles diffusing around, and we see a very high background. We are interested in the signal that is originating from these particles. And so we see that obviously this illumination technique is not really optimal. So what can be done? What can be done to improve this? And that is to use a very old idea, an idea which was actually brought up more than 100 years ago, and that is by focal plane illumination. Here in this sketch, you see that the illumination is not occurring into the sample from the top, and the signal is not detected along the same axis. But now we illuminate the sample from the side and illuminate only the focal plane. And that has a big advantage that we don't excite out of focus fluorescence, but only the focal area, which then an effect which greatly reduces improves the contrast. So this idea was brought up by Zsigmondy about 100 years ago. It was taken up by a French working group, Voyer et al. In 1993. And it was especially revived by the working group of Ernst Stelzer and his co-worker Jan Huisken. Since 2004, when that key publication has appeared, the technique really was developed in a lot of different directions, and it's still going on, and new ideas and new applications and new ideas to improve contrast and resolution are still occurring. So I will show you the effect that is achieved by that side illumination. We saw this before, but now when you use only that illumination from the side, you see that the contrast is greatly improved and that you easily can make out single molecules diffusing in that bulk solution. Okay, so often we use not that type of illumination that was used in this experiment, but we use an illumination from the side using a scanned laser beam. So in normal fluorescence, as I said, we have the illumination and detection through one objective lens. And now in light sheet microscopy, we often use the so-called digitally scanned light sheet microscopy which works like that we scan a laser beam along one plane and we illuminate our sample using that laser beam. And fluorescence is detected then perpendicular to the illumination direction, either in top direction or bottom direction. So this is the principal idea, which then can be refined to achieve new features. So the basics of the instrument still is usually more or less principally the same. So I show you here a sketch of our instruments that we built in 2008. You can see here on the left-hand side the illumination lasers. You can use, of course, various colors to illuminate your sample. They are brought together by certain spectrally sensitive mirrors, and you illuminate the samples principally from the side. The sample is located here in usually a liquid surrounding. That liquid can have different chemical features, but the key point is that then the detection is done perpendicular to the illumination direction and then achieved by a CCD camera. Usually we illuminate the sample, and while scanning the beam, as I showed before, along that illumination plane, we record one image with a CCD or nowadays with a scientific CMOS camera. When we compare this approach to improve the contrast with the probably more well-known confocal laser scanning microscopy type, we can see the following. From confocal microscopy, we illuminate an extended sample by a focused laser beam, which is guided in a plane along the sample. We illuminate it in one direction and we scan along one line and then the complete plane. And correspondingly, we detect the fluorescence emission at the respectively excited points using the confocal pinhole. So we have a serial detection and a process which necessarily takes some time. In light sheet microscopy, we illuminate the complete sample plane, sometimes in one instant using a cylindrical optics that illuminates the complete sheet in one instant, or using the scanned type where we scan along the plane. In both cases, we will detect the fluorescence in a parallel manner using a camera. So what are the consequences of these different illumination and detection types? In confocal laser scanning microscopy, we have a relatively low frame rate because we need to address each pixel separately. We have a relatively high photo damage because we have to illuminate the complete sample from bottom to top whenever we scan one plane. We always have out of focus fluorescence excitation, but the good part is, of course, that we remove that out of focus light by our thunderstorm. I just go and close the window. So I hope everything goes well and we don't have further problems. Whereas with light sheet microscopy, on the other hand, we saw that we have a high frame rate because we can detect a complete sample section in one instant using a parallel light detection device. We have very low photo damage because we illuminate only the plane of illumination and the plane of detection. We have therefore no out of focus excitation, and we have, but this is a disadvantage, of course, we have the detection of scattered light, which might reduce the contrast. However, there are two ways to get around this part of this disadvantage of the scattered light. One is to use confocal laser scanning, confocal light sheet microscopy, which we introduced in 2012, where we demonstrated that using confocal line detection, you can improve the contrast of light sheet microscopy even further. And the other one is by expanding the sample, and we come to that in a few moments. Here I show you a list of the instruments that we built in the course of the past years. So we started out using light sheet fluorescence microscopy for using single molecule detection, as I showed the example in the first slides. Then we introduced a confocal light sheet microscope. Our third instrument was that instrument that then reached Santiago de Chile, and we did that in the collaboration with Steffen Hertel, who I already saw in the audience, and Miguel Concha from the Universidad de Chile. And here we see the two students that actually built that instrument here in Bonn, and then it was taken apart and transferred to Chile and set up there again. So that was a great enterprise and great fun. Okay, after that instrument, we built more confocal light sheet microscopes. We built one for the Institute of Reconstructive Neurobiology in the University Hospital in Bonn, another one for the Institute of Anatomy. And then we have developed 4 further instrument types in my working group here. And of these instruments, these ones are still operational. The experiments, which Juan will later demonstrate, have been done with this final one with our high-resolution light sheet fluorescence microscope. Currently, we also work on illumination using the Aero beam. That is another student of mine working that. So what is the advantage that light sheet fluorescence microscopy provides really for the study of neurobiological samples? This I want to show in this slide. So looking at a complete brain, like here on the left-hand side, we see actually a light sheet fluorescence image of a complete mouse brain section. Actually, the sample was complete. It was not a section, but this is a light sheet section. And you see that sample is relatively large, about 1 centimeter in each direction. And now the key question is, looking at such a sample, how can we really tell in what way and how neuronal connections are established from one position to any other direction? So what we need to answer that question is, on the one hand, the capability to image a complete mouse brain, or at least sections, thick sections of mouse brains, and on the other hand, achieve very high resolution that is so high that we can see spines and synapses, because these occur at length scales of tens of nanometers only, and we must be able to reach such a resolution in an instrument that is capable to image complete mouse brains as a whole, or organoids or something like that. So that contradiction, these demands can only be fulfilled by a light sheet fluorescence microscopy that we combine with expansion microscopy in order to achieve the necessary resolution. So we will answer that question, how to achieve the detailed imaging of neuronal connections in mouse brains and organoids ranging from the nanometer to the centimeter scale. Okay, in order to achieve a particular optical resolution, we need to tell apart the fluorescence from 2 points of interest, as indicated here. So classical microscopy has its limits where these points are too close to each other, because if the objects are very close to each other, the respective point spread functions in the image plane will overlap. And this will not allow to discriminate such closely located points of interest from each other. So in the past 20 years, scientists developed several techniques in order to improve the resolution so that we can indeed resolve such closely related points. And that is usually done by the means to sharpen the point spread function using techniques like STED microscopy, structured illumination microscopy, or storm microscopy. And these physically improve the resolution. So this is one approach to achieve that goal. The other option is very simple, namely to expand the sample. So in that case, the points of interest by a physical expansion of the sample will obtain a distance from each other, which then allows to discriminate them easily and directly. So this approach is realized by expansion microscopy, and we combine expansion microscopy with light sheet microscopy. So how is that expansion brought about? I have here a sketch. We see here an example of a single cell and some proteins that are located in these cells. And what we are interested in is really to see the very details of these cellular or intracellular structures, which are shown magnified here in that upper part of that sketch. So we take such a sample, and the first step of expansion microscopy is a labeling. We do that either by autofluorescent proteins, by dyes like Höchst, or by antibodies in normal preparational approaches. So we achieve a sample which is then looking like that. In this example, we use immunofluorescence. So, and we see that these fluorescent antibodies are then located in the sample after permeabilization of the sample. In the next step, we do a chemical linking. Here we add a bifunctional linker that on the one hand attaches to protein residues in our sample, and that on the other hand allows to attach it to a gel structure, which we add in the next step. Here we actually form typically a polyacrylamide gel within our sample, and that means that we rigidify the complete preparation. Now in the next step, we digest the major part of all proteins. That means that the antibodies and all structures within the cells are preferentially lost. But since we keep some of the intracellular, the protein structures, the smaller peptide regions, and the dyes are still linked to these, and on the other hand, they are linked to the gel via the bifunctional linker, we keep the fluorescence in place. And if we have this sample by the digestion process, it is already quite transparent now. And if we keep it in phosphate buffered saline, we have usually already a magnification of about 1.5-fold. When we exchange that buffer by redistilled water, we usually have a 4-fold expansion process, and the whole sample simply is physically magnified. That technique was introduced in 2016 by the working group of Edward Boyden, and it really changed a lot in fluorescence microscopy since then. So the advantages of using this approach is that we first achieve a transparent sample, either in this way, about 1.5-fold expanded or 4-fold expanded. And that now the sample has the refractive index that is almost identical to that of water. Nowadays, new expansion techniques have been published, and so expansion factors up to 20 have been reached meanwhile. But we typically stick to the 4-fold expansion process. I will show you an example of that. So here, on the one hand, on the left-hand side, you see an organoid sample, which is about 2 millimeters in diameter, and it is already placed inside the gel. After digestion with protein case and exchanging the buffer medium to PBS, we have that mentioned 1.5-fold expansion, and you see that clearly in this image here. If we digest, take the digested sample and place it into be-distilled water, we have that 4-fold expansion, and you can directly see the size difference. And if we stain that sample, then we get images like that if we examine the sample with light sheet microscopy. The light sheet microscope, which will be used in the studies, and the images that Juan will show you, looks like this. It is actually a quite plain instrument, but the point is that we optimize everything. We optimize the filters, the illumination fields, the camera, the detection, the detection objectives especially, and by that means we achieve effective optical resolutions. Finally, if we take the magnification by a factor of 4 into account, then we achieve optical resolutions laterally of about 80 nanometers and laterally and axially of 300 nanometers. And this, I would like to stress, is 2-fold better than any confocal laser scanning microscope can achieve. We use, as you can see here, we use different objective lenses for detection. I would like to stress that in the nanoscale, so with very high resolution, we work with a 25x NA 1.1 water immersion lens. Whereas when we want to observe samples on the mesoscale, that means we want rather to achieve overviews, then we use a 10x NA 0.3 water immersion lens. Okay, with this introduction to the technique and to the instrumentation, I would like to hand over to Juan, and I stop here.
Okay, I hope that you can see my presentation. So thanks, Uli, and thanks, Alenka, for the invitation and introduction. I will start, I will show you 2 examples or 2 applications. The first one will be with human brain organoids. Now it's working. So as Uli mentioned, so we have this expansion factor up to 4 times, and in the bottom we have like a general sketch of what we are doing with these human induced pluripotent stem cells based organoids. So first we permeabilize the organoid to avoid a good antibody staining across the complete body, and then becomes this light sheet fluorescent expansion microscopy technique, like embedding this polyacrylamide gel, digesting, that means removing all the lipids and breaking all the proteins inside the sample, to choose later between a 1.5 time expansion or a 4 times expansion, depending which buffer do we use. So with that, we could use exactly the same sample and image that at different scales, like mesoscale, microscale, or nanoscale. So that is important because now there is no need to cut the sample and transport the sample to a different microscope in order to get a different kind of resolution. So for the mesoscale, I will show you here a video where you can see a 2-month-old brain organoid that is that have like nuclear staining like hogs, and the time junction marker Z01 that is marking these ventricular zones inside the organoid. So with the nuclear staining, we can get the volume, and with the other one, we can get the number of rosettes that are in the within the organoid. So I don't know if you can see my mouse. So here is a close-up of one of these rosette structures where the ventricular zone is stained. And what we are doing now is, with all this information, we can do a segmentation of all these bodies inside the organoid and get some numbers. For example, like the volume of the complete organoid. In figure D, you can see that the distribution of the apical surface in 3 different 2-month-old organoids, where we have about 480 of these rosettes or apical surface inside. So that's a quite nice example to show the potential that we have when imaging in mesoscale. So another example could be to see what is happening with these brain organoids during development. So here what we have are brain organoids that express GFP in 10% of the cells. And we have samples of 1, 3, 5, and 14-month-old. And in the first one, we can see that the cells are actually clustering in one part, but then we cannot get the same conclusion if we only look at optical slices. So if you work with 2D slices, you will not be able to conclude if the cluster is still there or if there is an even distribution of the cells in these samples. So that's why we now, with this technique, we have the ability to image the complete organoids. And for example, we see that the cluster actually remains along development, and we see only a little cells that are moving in the brain organoid, and that is probably due to the proliferation of these cells in time. Okay, so these stem cells start to divide and form new kind of neurons and cells inside, and they're moving to different positions. Now, if we go and we change objective and we go to the microscopic scale, what we can do is to analyze, for example, the cleavage plane in an organoid. So why are these important? Because there are like some neuronal disease that affect the incidence of these different cleavage planes. So in A, we can see the definition of the 3 types of cleavage plane: horizontal, oblique, and vertical. This refers to the angle that forms the division cell with the apical surface of the ventricular zone. So here in this part, we can see that we have the Z1 staining. There is the type junction that it's located in the center of this rosette structure. And in this dividing cell, we see that looks like it is a horizontal cleavage plane. But the problem when you only work with 2D slices or with 2D images is that you can misinterpret the data. And that's why in D and E, I'm showing you exactly the same cleavage plane, but from different angles. So the first one is XY, and then if rotate the sample and we look from the X and Z plane. So in one we have an horizontal plane and the other one an oblique. And that's why it's important that we can keep all the 3D information of this sample in order to be able to analyze properly this kind of subcellular structures, like I'm showing you in this video here. Right? So with that, we analyzed about 51 different cleavage planes in 3 different samples, and we found actually that the distribution of this cleavage plane was normal, like for a wild type human brain organoid. So that also was help us to support like these 2, right? And now, if we want to go to the nanoscale, right, then we will analyze here the pre and postsynaptic structures in a 14-month-old brain organoid. So Here is an optical section of a 3D volume, and this was done with 285 different single tiles that are stitched together. And we will focus in that area that is marked in A. So here we can see GFP expressing cells. Not all are the same kind of neurons because they were GFP expressing stem cells, right, that developed into something. But what we did is to use the pre and post-synaptic markers, Homer1 and synapsin1, to see if we could find synapses along these GFP positive cells. So here is a close-up of this region where we see the GFP in blue. So I'm showing in blue and because it's easier to see the colocalization of the pre and postsynaptic with red and green. And here we can see actually that we could colocalize perfectly when we do our 3D reconstruction. We can perfectly see the colocalization of this pre and postsynaptic protein, in this case in a multisynaptic button. With this data, then we are able to do the analysis of the synaptic distance in an organoid, where we got a mean value of 150 that correlates good with what is shown with other super resolution methods like STEP or STORM. But here I would like to point that still we have the complete organoid that we have, so we can have a lot more information, like when you use like this other super resolution method that have a limited working distance. So in this case, we could get more also from one single sample. Also with this, we are able to quantify like how many dendrites we have in some area. In this case, for a 14-month-old, we found that was about one dendrite every 200 square micrometer. Okay. So in summary, for the organoids, what we are able to do, so we can analyze the complete architecture of the organoid and the cell distribution. We can analyze during development. Also with the microscopic scale, we can go to and analyze the position and orientation of subcellular and cellular structures, and also the analysis of spines and pre and postsynaptic proteins that helps to identify like mature neuronal networks. Okay. So the next example is with mouse brain sections. In this case, what we wanted to see is a long connection that is between the HDV and the olfactory valve. Now I'm going in mouse brain sections, and these projections are really long, so are about 4 to 5 millimeters long in distance, and there is still not known like where the cells of the HDV are projecting into the olfactory valve, because the olfactory valve is made up to with different layers. Okay, so there is the glomerular layer, external plexiform layer, mitral cell layer, and granular cell layer inside that area. So what we did is try to cut this in a slice, in a thick slice that contains a complete olfactory valve, but also contains the complete HDV region. Okay, and for that we need to take a slice that is about 2 millimeters thick. Okay, to be able to apply the technique in this thick section, we have to optimize the protocol. And here's how the sample looks like when you embed the sample in the gel, and then when you have how the sample looks after the digestion process. So it's not as transparent as the brain organoid, but because this is also a more complex structure, but it's transparent in our areas of interest. Okay, this is the lower part of the brain, where is the HDV and the olfactory valve. And here is an example, right? A mesoscopic image of this mouse brain sample that was injected in the HDV area. Here we can see how where the injection site is and how the projections are going into the olfactory valve. What is nice about this technique or this optimized protocol is that we are able to retain the GFP fluorescent proteins. So we don't need to do an antibody stain. And that's really, really good, as we can see here in this video. Okay, where we see the nuclear staining, and we can see clearly the two main areas of the olfactory valve, like in the middle, this granular cell layer, and in the outer part, the glomerular cell layer. But we also see like some GFP projections there. Okay. If we zoom in even more here with a better objective or a high magnification objective, we can see that in this part we are actually full of these projections from the HDV. What is important is now to see where these projections are going and how are they interacting there. Right. So something good is that we can retain this endogenous fluorescence. Something maybe that is not that nice is the amount of data that you will generate with this kind of images. Okay. Now, to avoid any antibody staining and also to see the interaction of these cells in the olfactory valve, I mean, with interaction, I mean like where they are doing synapses, we inject new samples with a different virus that is expressing now GFP in the complete cell body, but it's also expressing mRuby in the synaptophysin terminals. Okay. So here is the test with Cree cells. I mean, we use Cree mouse in order to only target a specific group of cells. That's the positive and negative control. And there we can see what this virus will do. So we will have GFP expressing in the complete cell, and only in the synaptic terminals we will have red dots. Okay, so now the idea is to see if we can preserve after this long process or long sample preparation to preserve the mRuby signal. So we did this test with 3 different mice groups for cholinergic, GABAergic, and glutamatergic neurons that are the most important present in the HDV. And the first idea is to see where the projections are located. So what we are doing is, I have this, okay, now. So we have this complete volume, right? But now we will focus only in the olfactory valves with a higher magnification objective, and we will get a volume like the one that I'm showing you in the video. So here, what we have finally is all the information in a thick section or in a block of the olfactory valve. And the idea is to see how the neuronal projections are distributed in this, mainly in these 2 parts, like the glomerular that is outside or around the olfactory valve, and the granular cell layer in the middle. So for that, one way to do it is making a X profile. So in this case, we are taking a 3D profile along the X axis. So in green is the fluorescence intensity of GFP, and cyan is the nuclear stain. So with that information of the nuclear staining, we could identify where these layers are located. We can do the same, right, with this set profile in 3D along this along set. And here is maybe a bit more clear, like the layers, these 3 different layers. in this cholinergic neuron. And we did the same with the GABAergic just to show a difference, right? So we can see that for cholinergic neurons located in the HDV are sending most of the projections into the glomerular cell layer. And the GABAergic neurons, these inhibitory neurons, are sending most of their projections into the granular cell layer, like in the middle part. So one thing is to see where the projections are going, but now we need to see if what they are doing in or if the projection is actually doing something in that area. And for that, we will analyze now this synaptophysin, this presynaptic marker that is included in the same sample. So if we zoom in, right, we can see the location, like the colocalization of the mRuby with the GFP signal, but the resolution is not that good. So for that, we have to go to a higher resolution objective, and that is what I will show you here. So this is the same example that I showed you before, and that was acquired with a 12x objective. And now if we change to a 25, like in exactly the same sample and the same instrument, we can see clearly the difference between these two images. Okay, so unfortunately, I was not able to find exactly the same location, but here you can clearly see how we can go with the one sample from the mesoscale to the nanoscale again. And if it's necessary, nowadays we can go even further. So if we focus in this region of interest, market in B, so this is with the 25 objective that we have, and you can see nicely the neuronal projections, but maybe the colocalization, but not that precise. If we have a look with a 100x magnification objective, so in this case we have exactly the same area, and here we can see exactly where the synaptophysin proteins are located along this structure. So to summarize, so we have an optimized protocol for these thick samples. So 2 millimeter thick samples means that after expansion, these samples will be 8 millimeter thick. So that's something that is not that easy to put under a microscope, as you would guess. But what is nice is that we are also able to preserve these autofluorescent proteins, in this case GFP and mRuby. So we avoid the use of antibody staining, because again, a 2 millimeter thick sample will be a bit difficult to stain properly. It is possible, but we want to avoid that kind of procedure. And well, we are able to analyze the distribution of these projections, and thanks to the nanoscale resolution, we are also able to see how the distribution of the synaptic connectivity into the olfactory valve is, and then to compare the function of different group of cell bodies there. So finally, I would like to thank all our group here, especially Dr. Jan-Peter Sibrase. The mouse brain samples we got from the University Hospital in Bonn with Dr. Martin Schwarz from the epilepsy group, and the human brain organoids, we got that from the reconstructive neurobiology department of the University Hospital in Bonn. So that's all from my part. So thank you, and if you have any questions.
Thank you very much to both of you for such an informative seminar. I think there are some questions that have been being addressed in the chat. Let's see. How much antibody is necessary for a light sheet?
So what we are doing is actually normal antibody staining. So there is nothing special. We use commercial available antibodies. So if you do your staining and it looks good in the confocal, I'm sure that it will look good under the light sheet microscope. So, but you need to have one thing into consideration is that normally when you do the staining, you have like an amount of fluorophores, right? And you are illuminating that amount of fluorophores and with the confocal. But in expansion, these fluorophores are also being separated in space. So when you illuminate, you will have less intensity if you compare the same image in like before and after expansion. Okay, but that is okay, and it's basically because the fluorophores are now more separated, and when you illuminate one plane, you will have less fluorophores in that plane.
And that is, I mean, in order to stress that point, that is a factor of 64. Yeah, so that is a considerable effect that happens here, and that is simply due to the expansion in a factor of 4, and that in three dimensions. So, for example, we had problems when we looked at bacteria using staining, yeah, because bacteria anyway have only a low number of fluorescence and not many proteins that they contain. So if you expand these, then the samples become relatively dim, and the labeling is really key to success in that case.
So you haven't had to alter, but you alter the dilution then? That's the other part of the question, or you don't have to use?
No, I use the same concentration. I mean, maybe I will need to use more volume, but because I'm having the complete sample. So I use enough volume in order to cover my sample, but I'm not increasing concentration of antibodies.
If anybody has questions, please go ahead. I think you can unmute. Oh, here's one, Ignacio.
Yes, thank you, Lenka. Oh, very nice talk, Ulrich and Juan Eduardo. I'm not familiar with light sheet microscopy, so I'm wondering if it's always necessary to do this expansion of your sample.
Maybe I answer. Of course not. We do not always expand our samples. Actually, we set out using not expanded samples, and many people do that. Mm-hmm. And of course, if you want to examine live samples, an expansion will not work because you kill your samples. Mm-hmm. So, for example, in order to examine samples like fish, yeah, we have one instrument where we look at the development of zebrafish. In that case, in order to optimize the penetration of the light into the sample, we use a 2-photon excitation, but it works also with single photon, but then you might harm your sample. I mean, these are the normal cases, yeah. But expansion is not always required. Now, I really want to stress this. It really depends on what you are interested in.
Okay, and what is the advantage of expanding your sample?
I think the advantage, Juan, go to your first slide, and that shows it immediately. Yeah. Now the sample in image A, you see the white point. Yeah, this is a sample. It is scattering the light heavily. Yeah, and that will really reduce all information that you will get out of your sample. But in B and C, the sample is transparent. And so you have no light scattering. The penetration is much better of the light into the sample and out of the sample. So that is a big advantage. So I know many people that use just the expansion until B, and we often do that always, in order to reduce the scattering and to get the sort of like as a preparational step in order to improve the light penetration into the sample.
So if I'm working with brain organoids and I would like to do like imaging, do you think that because I cannot do either this expansion or clarity, you think that is a big problem to perform these experiments? Maybe doing light sheet microscopy is not the best idea?
Maybe with that 2-photon light sheet.
And up to a given size, it will work very nicely. I think this is like in normal microscopy also. You will get sooner or later get problems with light scattering in your sample, but you can look at live organoids during their development with light microscopy. It will definitely work better than confocal microscopy.
Okay. Okay.
Because the illumination is more benign. Yeah, you don't photo bleaching is reduced and everything. So it's certainly an excellent technique for that. We never tried it though, because our collaborators do the organoids and we do the imaging here in our lab.
Thank you very much.
I would like to add something. So there are also one advantage is of course that you have like a completely transparent sample when you use like expansion compared to other methods, but also that is you don't need any special objective because it's finally water what you're putting there, or PBS. So so that's one thing. So like for example, if you try to clear your sample with organic solvents, usually you need a special objective for that. And the other thing is with expansion, you are able to keep and like all these GFP, YFP, like a really big range of autofluorescent proteins, and also to use most of the commercial available secondary antibodies.
Okay.
So there is no restrictions with that.
Okay, thank you.
I see some more questions in the chat. So from Alex, he's asking, do you have experience expanding spheroids and how straightforward is it?
I mean, spheroids are usually made with in a similar way, like a brain organoid. So it's we test actually exactly the same protocol, and most of the images that I show you here, we have the same staining and everything with spheroids. So it's also possible to do.
Thank you. And there's another one from Raul Sepulveda. How many channels can you detect at the same time? Does it depend on the filter wheel?
In this case, I only detect one channel at the same time. No.
Yeah. Also we have another instrument where we have a dual channel adapter by Hamamatsu. And in that case, we can detect two channels simultaneously because you need a dichroic mirror in order to distribute the fluorescence into two channels, of course. And you can probably principally expand that by using more of such devices. But in view of the fact that one of them costs about 30,000 euros, you have to think about the costs of your instrument. But there is in principle no limit. So, but in order to extend that point, there is one advantage in using only one channel at a time, and that is the alienation of the images. Because in that case, I mean, you illuminate your sample and you detect the same region, and you can be sure that they are perfectly aligned. In case you use several cameras, you have to align your channels posteriorly. And that might produce problems. Yeah.
Yeah, Ignacio Velasquez is commenting that the system at Universidad Mayor can detect 2 simultaneous channels and more sequentially with proper alignment. Okay, another question from Raul is UC2, open and modular optical toolbox, has a light sheet fluorescence microscope. How good is it?
I don't know that one.
No, me neither.
No, can't comment. We have here an instrument in our lab now also from Miltenyi, the Blaze, yeah, and that we use for more traditional samples. It's a workhorse, so to say. Yeah, it's not as sensitive and not with such a high resolution as our home built instrument, but that's also valid and works very nicely, I would say. But the other ones we don't know. There comes a question of what the range of chromophores is. I mean, that is like in usual fluorescence. You can do absolutely the same. You need a laser to excite your fluorophore, and you need a filter system to detect it. So it's like in any confocal microscope. You can use UV dyes, you can use infrared dyes, so whatever you like.
Okay, Raul is asking, is an open source 3D printable microscope?
That is the UC2.
Oh, that's the UC2. That's open source. Okay.
Okay. No, we haven't tried.
Okay, from Jorge Toledo. Hello, Juan Eduardo, congratulations, nice work. I'd like to know if when you have very large samples and you rotate them to take photos on light sheet, does the expanded sample vibrate? Is it necessary to additionally fix the sample to give it more firmness?
So I don't rotate the sample to take the images. So usually the gel is laying on a cover slip. And as Uli showed in the setup, we have the illumination from the side and acquisition, like a upright acquisition. But what Jorge asked about, like to give it more firmness to don't have like a really like a fluffy gel. That's more of the sample preparation. So. So that maybe something is not being, something needs to be optimized in his case. Because I also saw that like with other, I have that experience with other samples, and usually that could be optimized, changing some concentrations and stuff. But I don't know which sample is he using.
I mean, you fix the samples to the cover slip using
with the
polylysine.
Yeah, just polylysine, so no glue at all. It's enough.
Well, I think, I don't know if there are any more questions, but it was a very wonderful talk. Thank you very much for presenting all your data, and we really look forward to working with you more and learning how to use these techniques. So thank you very much for a great seminar.
Yeah, I think it's not so easy to bring all the ideas over in such a presentation type. That's at least the feeling that I have now finally. So I'm looking forward, and I really hope that we can realize our visit in Santiago in January or so, and then we can get a much better contact and discussion rounds with people in presence, hopefully.
Yes.
Yeah, it will be really nice to make a hands-on with the technique.
Definitely, we're going to plan that very soon and make sure people mark their calendars probably sometime in January. So I think that's going to be great. We really look forward to that.
Last question.
I do see one more question from Ramon. How do you ensure antibody penetration to the whole sample?
Yeah, the permeabilization step is it's critical for that. So that's why we are using a different kind of detergent, not Triton. And yeah, so that's one thing. And other thing is that sometimes, in some cases, the antibody staining could be done after the digestion step. And that also improves penetration, but you have the risk to don't have your protein of interest anymore, because basically you are doing that. You're removing lipids and breaking proteins inside the sample. So that's something that needs to be tested, like depending which kind of marker do you want to use. But we usually try to do the antibody staining before, because then we are sure that we are targeting the specific protein. In the size, yeah, it is possible. You need depending how big is your sample, you should be able to keep the gel hanging from a cover slip that is coated with polylysine. So I think that I did some tests with something like that in another light sheet version that we had before, and yeah, but depends on the geometry of the sample, like especially if it's flat or like a cube, still it's possible to have the cover slip and the sample hanging here. Mm-hmm. Okay, and polylysine will be strong enough to hold that.
Okay, and do you use fiducial markers to do the merges in the light sheet? How do you anchor them to the gel?
Fiducial markers, I mean, it could be possible to use amine modified bits that if you want to have like a mark, something outside your body of interest, like outside your sample. I mean, you will have your sample, you can put some bits in the gel, and those will be usually outside. And then if those are like multicolor bits, like these tetraspec bits, then you could use those ones to align the volume.
But we usually do not use them because, yeah. I mean, we image the sample in several channels quickly, one after the other. So there's not really need to align things. Yeah.
Yeah. Yeah. In our case, it's like really precise. So we scan the sample and we have usually no problem with that.
And I think the size light sheet, they have a specific sample chamber where you can place also larger samples inside, I think. Yeah. I'm not completely aware with the last version of the system, but I think I mean, the things that we do can also be done with a Z1 light sheet, yes, as far as I know, at least.
I think so.
I think that the only limitation there is the how much distance do you have between the objectives. Mm-hmm. That's the limiting factor finally.
Right. So when you have your 8mm sample, you are not limited in your setup.
And with the working distance, depending the objective. If I want to go to the nanoscale part, then yes.
Okay.
Because I have like 2 millimeter working distance.
And in that case, would you slice
Then-
that?
Then we are working in a solution for that.
Okay. Okay. I think we have some information from Ignacio at Microx Chile about some mesoscale accessories for larger scale sizes.
Yeah.
Extra magnification.
Okay, well, again, thank you very much. We hope to continue these conversations. Yeah, we'll look forward to seeing you in the future.
Okay, thank you very much all for listening and discussing, and thank you very much, Alenka, for organizing and guiding us through this meeting here.
Of course, very welcome.
The same goes for my part. So thanks to everyone for listening and for the questions, and thanks Alenka for all the organization.
Okay.
And a special regards to all the friends that we see here being present. Hi, Victor.
And her. That's nice.
Okay. Yeah. Then see you soon.
Okay.
Yeah.
See you soon.
Have a nice day.
Have a nice day. Bye-bye.
Bye-bye.